Alright, welcome. So, we have paper 2 tomorrow, and what I want to do in this stream is I don't want to learn anything new. I don't want to, do a lot of big extended response questions. All I want to do is go over some generic notes for every single topic, really. I'm going to look at a past paper, just quickly see if there's anything I need to go over, but really, this is just going to be more of a chill last-minute stream. So, I hope all of you guys are well. I hope all of you guys are feeling good about this paper. Honestly, a lot of people found paper 1 difficult. I didn't. I did not find that paper difficult at all. I thought, yeah, there were some annoying questions, but none that were difficult. The hardest one, in my opinion, was talking about the structure of a leaf, because I completely forgot that was something we had to learn, and I never learnt it. So that was the only thing that stumped me, and I still probably got a mark or 2 from it. So I don't know where people were getting the idea that it was a bad paper from. I personally didn't see that, but look, if people found last paper difficult, maybe that means that the next paper is going to be nice. I don't know. So, one thing I want to go over, and this is really good to know, it's helped me a lot, is the idea of the evaluation questions. The questions were, you will be given a graph, a table of data, something like that, and you have to evaluate it. So, for example, again, I'm going to completely make this up, to be honest. Let's say we have, a bar chart, like this, and hello, hope you're doing good. So, for the evaluation questions, let's say you have a bar chart. The one thing that a lot of people forget, and I also forget it a lot, in fact, I've probably forgotten how to explain it now. If the standard deviations overlap, that is something that you need to talk about. Equally, if they don't overlap, you also need to talk about that. The standard deviation error bars, because that shows you whether or not the data is statistically significant. That is a very obvious thing, but I actually miss that quite a lot. I know a lot of people probably will. I've just drawn my pen. I should also add, because there's a few people watching, I'm not a teacher, I'm a student as well. I literally have no idea what I'm doing, okay? Please don't watch this as actual revision. So yeah, that is one massive thing. Another thing which a lot of people forget is how easy it is to get a mark for this question if you literally just mention that there has been no statistical test. So, if you're given... okay, you know what, I'll look for an example quickly. I will look for an example to explain this. Biology papers. I'll go to the 2023 paper. I'll take a quick look. Evaluates. Okay, yeah. This question here from the 2023 paper. Use all the information, evaluate the effectiveness of this. This is such a crude way of doing it, I know. It's a bar chart with standard deviation error bars. That is a lovely question. That is a lovely question. You could obviously talk about the error bars not overlapping. And usually it gives you, for these evaluation questions, a load of unnecessary information, but some of it will be useful. For example, here, it's showing that all of these plants are of the same species. And in the question, it's talking about all plants. So obviously that would be a point to talk about, is they've only used one species. Like, there's a load of generic things you can say to get extra marks that, like, really are very obvious, but you can easily miss out on. So, standard deviation, error bars, the idea of have they done a statistical test or not, the idea of have they done it with multiple species or only one. 9 times out of 10, they've only done it with one species, and therefore you can't make a claim about it being for all. The idea of a sample size. Now, this probably isn't a frequent mark point, but if they've done a sample of like 10 mice, then clearly that's not going to be representative of the data. Also, that is another thing. If they've tested it on something like mice, and they want you to evaluate whether it will be good for humans, mention that. Mention that you can't make a valid conclusion. So that is just a kind of generic overview of the evaluation questions, because I don't think we got any in paper 1, as far as I can remember. I've blocked out paper 1, to be honest. I don't remember getting a question about having to evaluate anything, so I'm telling you now there will likely be 1 or 2 questions on it, which would be lovely, because it's 3 marks. Usually they're very, like, bulky questions to get marks from. Obviously, paper 2 has the reading comprehension question, which, like, there's no definitive way to answer this, really. You just have to kind of hope that it's something nice. Like, if it's a question on DNA probes, you might as well, like, you know, count your blessings. A lot of the calculation questions for paper 2, I personally find more difficult. Don't know about you guys, but I personally find them a lot more difficult. I thought the paper 1 calculations were really nice, but yeah. So what I want to do is... Hello. What I want to do is literally just kind of go over every topic quickly and just see if there's anything that I really need to, like, revise. I say revise as if I don't have the exam in, like, 13 hours, but yeah. I think, in my personal opinion, the energy transfers topic is the nicest. Praying for photosynthesis questions, because supposedly they weren't on last year's paper. I think we're all good with the light-dependent reaction and the Calvin cycle. Respiration would be a nice one. Things like the nitrogen cycle, I don't know how prevalent that is in the papers, but that would be a nice one too. Like, let me have a look actually. Nitrogen. Again, for those clicking on, I'm literally just, like, waffling here. I'm just kind of waffling about paper 2. There are much better people to watch than me, I promise. Okay, hold on. What is the 2021 paper? Have I gone over that yet? I have not. Okay. So, what are you teaching? Why do we have 8 people watching? Oh my god, hello. I am not a teacher. I'm literally just waffling about paper 2. Hope all you guys are good. So yeah, question on... Like, this is question 1 for 2021. Describe a process of glycolysis. That will be lovely. That will be a lovely question. The thing that I find that, like, I don't know how many of you guys do this. What grade? This is, this would be, like, A-level biology. year 13 content. The process of glycolysis, that would be a lovely question. AQA. The process of glycolysis would be a lovely question. The thing that I don't know how many people do this. I always write in bullet points for like these extended response questions, these 4 to 6 markers. I don't know how many people do that, but like it is a lovely thing to do. Like the question about glycolysis, you might be inclined to want to write like a whole page of information, but like generally speaking, you can literally get marks for saying like, you know, there is a net, literally just saying net gain of ATP would be worth a mark, like obviously, etc, etc. Again, I'm just kind of flicking through the papers, trying to give any advice I can. I really hope we don't get a question on IAA and like auxins. That would be an unfortunate one. Because the practicals that they usually show for that are like really gimmicky. Things about it being produced in the tip, IAA being produced in the tip, diffusion. Let's see. Oh, dihybrid crosses. Dihybrid crosses aren't bad. I would not mind a question on that. Again, I don't want to delve into showing anything specific here, just because, like, you know. I have to go over something. What should I go over? What do I go over? Okay. I learned that in 10th. Okay. Man, there are, I don't know why so many people, I mean, I know why people are watching. Guys, I promise, I'm not a teacher. I don't know anything I'm talking about. Please watch someone who knows what they're talking about, please. Okay, the one thing that I would not like is anything to do with gene technologies. I will go over this quickly because I have no idea about this. Things like the polymerase chain reaction. I do not like this at all. But I don't think it's that bad. Like, from what it says, you literally just have the DNA fragments. Obviously, the enzyme DNA polymerase. This is basically just explaining what... Oh yeah, thank you, man. I appreciate it. Take care. Obviously, DNA polymerase and nucleotides and primers. Literally, see, this doesn't sound that bad now that I'm... This doesn't sound that bad now that I'm saying it, but you literally just heat it to break the hydrogen bonds. You reduce it so that the primers bind. And then you just increase it so that DNA polymerase joins the strands. That's not particularly bad. That is not a particularly bad question for 4 marks. Literally just explaining what you need, which, like, is obvious. You have your DNA fragments, you have the polymerase enzyme to obviously join together the phosphodiester bonds, and nucleotide, like that's fine. That is literally just explaining what DNA replication is, just with a little bit extra. And then you can elaborate on the PCR reaction when talking about gel electrophoresis. I hate this one so much. You basically... This one is mainly due to the gimmick of the variable number tandem regions, which is basically just like obscenely long repeating like base sequences. That one is literally where you do the PCR on these regions, so you get DNA like it's the PCR, by the way, is literally just amplifying DNA fragments. That's what it does. You get like one DNA fragment, you amplify it, you get more. That's the purpose of this. So you do this reaction to get said amplified fragments, and then actually it's not that bad from there. Again, I seem to be misremembering how bad most of these are. You then get it on a slab of gel with an electrical current passed through. DNA has a net negative charge, therefore it's going to move towards the positive, which gets you like these cool bands. And it is literally that the lighter the bands, the further they are going to move. And obviously then you're able to compare the different, tandem regions, because if they are genetically similar, they're going to have, you're going to be able to compare and identify the same regions. Again, I don't know how many questions you could even get asked in relation to this. I've seen questions where you're asked to draw this. I've seen questions where you're being asked to compare. I've seen questions where they talk about, what would it be called? Splicers, I think. Oh, I don't know what you call them, actually. The things that, like, cut the genes. I've seen, like, the plasmids. I've seen questions where it will show you a linear strand and a circular strand, and it will basically ask you how cutting it will change the number of strands you get. So if you cut it 4 times in a circle, you're going to get 4. If you cut it 4 times in a line, you're going to get a different amount. What else? DNA probes I don't think is bad. It's literally, you get your DNA... Okay, I'm going to have to get rid of half of this now. Man, this looks like such waffle. Okay, the DNA sample is going to be broken down into DNA fragments and separated using gel electrophoresis. So literally what we've just said. Then, the fragments are going to be transferred to a membrane with the DNA probes. The probes are going to bind only to their complementary sequence. And usually, these probes are going to have like some indicator, such as like a fluorescent, like a UV indicator. Therefore, when they bind, you're going to be able to tell. So, a vast majority of the gene technologies, you can kind of just waffle, because most of them are really similar. Role of restriction endonucleases. It's literally just, they cut the hydrogen bonds. Then they form the sticky ends, because you will have, again, I don't know really how to draw this. If we assume these are like bases, it's going to be things like this, where you have these bits like that. they have the exposed nucleotides. Then different fragments can be joined together so that they're complementary. Like, that's literally it. Restriction endonuclease literally just breaks the hydrogen bonds. That's all you really need to know to then be able to write about it. cDNA and reverse transcriptase, this is literally just if we have, like, I don't know, some, like, pancreatic cell that produces insulin, then what you can do... Hold on, am I looking at the right bit here? Yes, I am. Obviously, that cell is going to be producing a load of mRNA because, you know, translation. Therefore, you literally just... All reverse transcriptases, all it wants you to say is you literally just add reverse transcriptase, and it's just going to create DNA from that. That is all you need to know. So again, I don't think that this topic should actually be too bad. Even if you get asked a question on, like, you know, the role of, like, I don't know, the role of, like, amplifying DNA or something like that. Like, it should be fine. Translation control is literally just where you have double-stranded siRNA. It associates with a splicer, so it becomes single-stranded. That's like 2 mark points, just by saying that. Then the single-stranded is going to bind to the mRNA, and it's going to cut the mRNA into small fragments, so it can't be translated. That's it. I think arguably one of the hardest ones is transcription control. I was talking to Olivia about this. I think we both find this one relatively difficult. This one is literally where you have something like estrogen. Because it's a hormone, it's able to diffuse through the cell surface membrane. The way this one works is it binds to... It's not a oh my god, I can't speak. It's not a transcription factor, it activates a transcription factor. So it binds to a binding site on the transcription factor, which alters its shape so that it can then go on to bind to the DNA. So, if you were asked a question specifically about that, or another hormone, it is not a transcription factor. It activates a transcription factor, and the way it does that, literally just by altering its shape so it can bind. So that's everything to do with gene technologies. I will go over also a really boring one. Genetics, populations, and ecosystems. This is such a boring one. But there's a lot of marks that can kind of be obtained here. Obviously, you have the different types of speciation. Allopatric and sympatric. I hope I said that right. Literally, this is just where one of them is geographic isolation, one is reproductive isolation. They are basically the same thing, though, like, in terms of what you have to say. Geographic isolation will lead to reproductive isolation. Because, yeah. For both of them, this cuts off the gene flow. There's no gene flow between the same species. Then you have your variation due to random mutation. Obviously, the advantageous alleles are going to be passed on, thus a new species is formed. And it's literally the exact same thing for breeding isolation, reproductive isolation. It's literally the same thing. Different alleles are passed on, and eventually a new species is formed. The only difference is geographic occurs, well, reproductive occurs in the same environment, geographic does not. That's literally the only difference. And usually it's worth like 4 marks just to talk about this. Literally the only other 2 I have here is inter and intra-specific. Inter-specific is competition between different species. That's literally it. Buddy, what the... No, that's what James did, man. What the fuck? I don't know why he was... I don't know whether anyone else's school would do this. If you show up late to an exam, surely you just get kicked out? That was literally it, bro. He literally just showed up like 25 minutes late to the exam. Like, while everyone else was in the exam hall, he just fucking shows up 25 minutes late. Anyways, interspecific is competition between different species. Intra is between the same. That is literally the only difference. Bro, I don't even know, man. I don't even know. So that is literally half of everything. The bulkiest topic by far is organisms respond to changes in environments. This is a bulky topic, and I feel like we're going to get asked the most marks about this one, I personally think. Things like the action potential, response to stimuli, myogenic control of the cardiac cycle, non-myogenic control, synapses, muscle contraction, the second messenger model, osmoregulation. I don't have time to go over all of that, but we will go over a handful. The action potential. This is where you have your resting potential of minus 65 millivolts, and it's maintained in 3 ways. The number one thing that you can say to easily get a mark is literally that this is more permeable to potassium ions, less permeable to sodium ions. That is like the number one marking point. The way that this is maintained is obviously you have the sodium ions being pumped out. It's 3 sodium ions pumped out for every 2 potassium ions pumped in. Obviously that's by active transport. And therefore this maintains a negative, this is not OCR, it's AQA. Obviously that maintains the negative polarization. The other thing you have is the leaky potassium channels, ensuring that this gradient is maintained. And obviously the sodium channels are going to be closed, therefore ensuring that this charge maintains. So that's the resting stage. And again, you can easily get marks for that. Like, you can literally get, like, 2 marks just for talking about that stage. Then it's depolarization, where literally, I mean, hold on, I don't remember what the graph looks like, actually. It's something like, I think it's literally that, where this is obviously a bit lower. Where you have, like, the all or nothing principle. So some... Oh my god, 12 people watching is crazy. Some of the sodium channels are going to open, obviously allowing sodium ions to diffuse in. If the stimuli is large enough, then the charge is going to exceed the threshold. That's where you get the all or nothing principle. And all sodium channels are going to open, raising the charge to 40 millivolts. From there, obviously, you have your action potential now. And then it's the hyperpolarization. This is where all of the potassium channels will open. This will rapidly lower the charge because all of the potassium ions are gonna diffuse out. And the important thing to mention, Matt, the thing is, a lot of paper one, at least from what I remember, was very much like very much like knowledge stuff. I'm convinced that we're probably going to get asked a lot more things like evaluation questions and actual application. I'm convinced anyways, but I'm not sure. Because obviously, like, all of us are going to be more confident with knowledge stuff, but I feel like paper one was very heavy in knowledge. I just completely forgot where the potassium channels will remain open longer than necessary. This is to obviously ensure that another one can occur. Very quickly, because this is something that I completely forgot, because I'm forgetful, the idea of myelination versus non-myelination. This is, like, quite an important thing to talk about. Where the action potential is able to jump between the nodes. That would only be worth 1 mark. So, literally saying that. The other, like, point, which sounds obvious, but, like, I feel like not many people would say, is literally the idea that this provides insulation. Literally, that would be worth a mark, just saying that. And then also, being able to compare this, the idea that in non-myelinated, the action potential has to occur across the entire length of the axon, in comparison to myelination, where it just jumps across the nodes. Because again, that is something they love to ask, is just being able to compare specific examples. I don't think we need to go over the Pacinian corpuscle. Photoreceptors, I'm praying doesn't come up. Aren't A level? No. I still have a handful more left. I don't think I can be bothered to go over photoreceptors. Like, if it comes up, it comes up, and I accept that, you know, I'm fucked, but it's not that bad, I'm just lazy. Myogenic control of a cardiac cycle. This is the nice-ish thing, where it is literally talking about the sinoatrial node, the wave of excitation. You can just link it to the cardiac cycle. Oh, you finish on the 9th? Man. That is lucky. I don't finish till, like, the 18th. Basically, if you know the cardiac cycle... Oh, no, I would hate that. Muscle contraction is the one thing that, like, I could ramble about, but I would not be good at. That is the one where the action potential is passed through the T-tubules, the sarcoplasm, role of calcium ions. It's the sliding filament bit that I don't get myself. Hello. Literally, I'm just waffling about biology to try and ease the tensions I have for this paper. I hope it doesn't come up. The thing is, I don't know whether they've ever asked a question on the entire sliding filament hypothesis. From what I was looking at, they seem to only either ever focus on the arrival of the action potential and the role of calcium ions, or they only ever seem to focus on the ending bit, you know, where the heads attach, the role of ATP causing them to bend, and the, what do you call it? The what do you call it? The moving. The power stroke, that's it. Like, I don't think they've ever asked a question where you need to refer to the entire process all at once, which is good. Oh, one thing I forgot is the difference between slow and fast twitch muscle fibers. I forgot about that. I want to see if... I literally, I can't remember this one. Obviously, fast twitch are like the more powerful ones. Whereas slow needs that like energy because it needs to be able to maintain contraction for a long period of time. I don't recall the differences in terms of structure of these. I know that one is going to contain more anaerobic compounds. I'm assuming that's going to be fast twitch. I'm assuming. Because obviously, you know, anaerobic respiration, that's why they tire easily. Because then it's the production of lactic acid. So they're going to contain things like glycogen, because glycogen can easily be hydrolyzed into glucose for glycolysis. So they're going to contain more anaerobic enzymes. Slow twitch, I don't remember what these have. I mean, obviously they're going to contain more like aerobic enzymes. I don't know what else they're going to contain. One of them I know contains creatine. Faster, more intense. Okay. That's good. You know what I might do? This is AQA. I'm going to pull up the chat. To be completely honest, guys, I did not expect this many people to show up, so I'm honestly just waffling. I'll go check the chat quickly, because I don't want to... Okay, yes. Okay, I think that should be fine then, because, like, realistically, they're not going to ask you 6 marks about this. Like, you know. The most I've seen is, like, a 4 mark where you have to compare both of them. They won't ask you 6 marks. I mean, come back and, you know, burn me at the stake if I'm wrong, but I really doubt they would ask that many. So I feel like just being able to talk about the idea of anaerobic, aerobic, and then the different compounds, I think that should be fine. Again, I hope. Oh, well, that is an alarm. Okay, I've just checked. 81 people have seen this stream. That is terrifying. Youch. Okay. So, that should be fine then. And I'm struggling to remember what else is on the muscle contraction topic. I know there's that whole thing of the Z-band and the A-band and stuff like that. I can't wrap my head around that at all. I will be completely honest. I've never been able to wrap my head around that. I know one of them's obviously the actin, one of them's the actin and the myosin. And talking about like the differences and how they expand and increase in size, that is something I've never understood. Like, if that comes up, I'll do my best to improvise, but like, I do not get that one, to be honest. The second messenger model, that is relatively nice. To be honest, that is relatively nice. That is literally just something will bind to the receptor, it activates something. But they can ask that in a lot of different contexts. No, I'm not a teacher. I am not. I don't know what I'm on about. They can ask that in a lot of different contexts. I've seen them talk about the second messenger model in terms of adrenaline, but they've also talked about it in terms of glucagon, things like that. The effect of the second messenger model is that it honestly depends. It honestly depends. Like, for example, one of them might activate vesicles, which will fuse with the membrane, which will put more glucose channels in the membrane, making it more permeable. Like, that's just a bit of a generic example. Ultrafiltration and the whole thing of osmoregulation, I think this is a lovely, like, topic. I think it is. Ultrafiltration is literally just the same as tissue fluid. It's basically the same. You have the high hydrostatic pressure in the... I don't remember what you call it. I think it's like the afferents arteriole. That literally is just going to force out substances, such as water, ions, urea, etc., out of the capillary pores. They will then enter the kidney nephron through the Bowman's capsule. Then it's selective reabsorption, which is literally just co-transport, but just worded slightly different. I mean, it is a form of co-transport, but it's literally, it's the exact same thing. It's sodium ions being pumped out, concentration gradients, sodium ions will enter through facilitated diffusion, bringing glucose with it. Glucose then diffuses out. It is a lovely, like, getting asked a question about would be lovely, because even if you don't know what you're talking about, it's just co-transport. Then osmoregulation, which is a bit of like, I wouldn't say it's a nasty one, it's just kind of a little bit dull to remember. It's basically just changes in the water potential that occur across the length of the loop of Henle and into the distal convoluted tubule. The way I have it written down, and again, I don't know how accurate this will be for the mark scheme, the way I have it down is the ions will be actively transported out. Remember, the idea is that one side is permeable to water, one side is permeable to ions in the loop of Henle. Water will move out of the descending limb. Obviously, hold on, I've missed a tiny bit there. The ions will get pumped out. This obviously will lower the water potential in here. The fluid that I forget the name of. Because the ions are there now. So that lowers the water potential. Therefore, water is going to move out via osmosis. It's literally just the idea that water is pulled out. That's literally the whole idea with osmoregulation. Water is drawn out. So water moves out. Then, because of all this water moving out, this filtrate is now going to be very concentrated. Therefore, ions are going to move out again via facilitated diffusion. And then obviously it does the same again. The filtrate is going to have a high water potential. Water is going to move out. It literally is. It's just playing with water potential gradients to get as much water out as possible using ions. So yeah, filtrate is very concentrated. Water moves out. The remaining fluid will move down the collection duct into the bladder, and the interstitial fluid will obviously enter the bloodstream. Then obviously we have ADH and the effect that that has on osmoregulation. I think this is also a lovely question. This is for AQA. But honestly, I'm not like teaching. I'm not a teacher. I'm literally just rambling. The effect of ADH. This is where you have the decrease in water potential, which is detected. This is assuming that we're going to have it be more permeable to water. ADH is secreted. ADH will bind to the receptor, so it's the collection duct, which, like, I'm just going to draw this, like, insanely crudely. It's going to bind to the receptor on the collection duct cells. It's active phosphorylase. Apparently I can't spell now. It activates active phosphorylase, and then it's the idea of aquaporins. These will fuse with the cell surface membrane, increasing the permeability. And that's literally it. That is literally it. Yeah, of course. Buddy, what are you on about? Yeah, actually, I'm not teaching a lesson, bro. I'm rambling. I am rambling about biology, and apparently we've had almost 200 people watch. That is scary. I'm doing good, man. Honestly, I'm just hoping that this paper's nice, but we'll see what happens. Okay. Module 6. Which one is that exactly? Because I just have the names here. Is that the organism's response to changes in environment? I think the easiest one is energy transfers. Photosynthesis, respiration, nitrogen phosphorus cycle. That it's lovely. Can you teach me? Honestly, I have no idea. 2020 live stream. Bro, it is. Yes, okay. Honestly, that's not the worst one in my opinion. I think the worst one's, like, the gene technologies. I just can't, like... It's boring to explain, and it's, like, annoying to explain. Even though I know it makes up a good majority of the paper, it would be an annoying one to have to yap about. I love talking about photosynthesis, glycolysis, link reaction. But they probably won't ever ask a question on the link reaction. I feel like that one gets neglected, but... Thing is with, like, genetic fingerprinting is, like, you can kind of improv it. Like, a lot of it is just using your knowledge of things like DNA replication and transcription, translation, but it is just a bit annoying. Oh, thank you, man. I appreciate it. Honestly, I'm just, like... I'm not a teacher by any means. I'm not qualified to explain any of this. I'm just kind of rambling, and I appreciate that you guys seem to be enjoying. Again though, like, please, if you're like panicking last minute for exam, like, by all means don't watch this. Watch someone who's qualified. I'm probably adding to the fear here by rambling about stuff I don't know. Do I quickly go over, like, glycolysis and stuff? I guess. I'm gonna, like, draw the worst diagram you've ever seen in your life. Glycolysis is lovely, because it quite literally is just glucose, then it becomes phosphorylated, and that's worth a mark. What did you get? I have not gotten results back yet. We're still doing it. When we did AS level, I got an A, but, like, I'd consider that paper, like, pretty mid. Yes, honestly, I'm scared of the required practicals. They scare me. The photosynthesis practicals are lovely. Thank you, thank you. That's the thing, actually. You know what? Forget talking about glycolysis. I'll go over the practicals quickly. You for unlimited marks. The DCPIP one, that honestly isn't too bad from what I remember. I say that because we literally went over it like 2 days ago in class. That's the one where DCPIP, it acts as the electron acceptor. And because of that, it will have a colour change the more photosynthesis occurs. I think the nicest practical is chromatography. All they really can ask you for this one is generic stuff, like, why would you draw the baseline with pencil? Stuff like how you would actually perform it, you know, but you have, you make sure it's slightly above the solvent front. You stop the practical once it reaches the top. You draw a line where it happened. Like, that's literally all they can really ask. I'll say that, and then tomorrow they're gonna ask some nasty shit about it, but... I think even calculating the way it moves, I don't recall that being a difficult equation by any means. I think that's literally just basic division. That one should be fine. I want to look at the other practicals. They ask a lot the respiration practical, so I'm inclined to say that won't come up. I'm not... again, I can't guarantee... but I have a strong feeling that won't come up. What else? Respiration in single-celled organisms. Yeah, I don't think that's going to come up. Dehydrogenase activity in chloroplasts. I feel like, yeah, strong potential. Simple animal responses. That one would be... I don't know, actually. That one would be annoying to explain. This is where you have kinesis, which... that is the random one, where the organism just moves all around. And taxis, where it moves specifically towards the stimuli. So, for example, if we've split this in half, and this is like 35 degrees, this is 25, it's going to move towards the favourable side. That one shouldn't be bad. I've seen a lot of nasty questions about organism responses. I can't lie. Like, there was one about, like, termites, I think, winged termites. That was an abysmal question. So I don't know. That one might be a bit 50-50. The effect of different variables on species distribution. Wait, what is this one? Is that quadrats? Oh yeah, okay. Please be a quadrat question. That's the loveliest practical, like, you could ever do. That is literally where, for 1 mark, you talk about the idea of randomly obtaining coordinates. If we have, like, a 200 by 200 field, randomly generate coordinates. If you get asked why, it is literally just to remove bias. So you're not picking, you know, your own. You would use a large sample. They want you to say that you use a sample of over 20 most of the time. Just say a large sample, because then it removes anomalous results, and it becomes more representative of the whole area. Then you can get asked 3 types of quadrat questions. You can either get asked the percentage cover, estimating the actual like number of the species, or you can get asked just the overall, I forget what you call it, like the frequency density, which is literally just basic addition, basic division and multiplication, sorry. So the quadrat practical is lovely. That would be an amazing one. Chromatography would be a lovely one. Respiration in single-celled organisms, I feel like that one won't come up. If it does, even that one's not that bad. Talking about, you have like the organism here, you have like a tube with liquid in. If it moves closer, that's going to be because the pressure is reduced, I think. In fact, you know what, I always get this mixed up. If it moves to the right side, that's because the pressure increases. The pressure will get reduced when obviously oxygen is drawn in by the organism. The pressure is going to decrease when... No, sorry, have I said that the wrong way around? Pressure will increase when there's more gas being released. Pressure will decrease when there's more being... my bad. You can tell I'm getting tired, because, like... Man, it is sickening that I've had to have a, It is sickening that I've had a history exam today. I am so tired. I am so tired. Okay. We've been live for nearly an hour, that's mad. I'm happy to keep going and just waffle for however long, honestly. I'm just like... Okay, the animal response is one that should be fine. Yes, there was maths. What spec? This is AQA. I know Declan did A-level math today. He said it went okay. He said half of the paper was shit, but supposedly the bit on mechanics wasn't the best, but I cannot confirm or deny. I was not there. Let's have a quick look. OCR, no, this is AQA. Declan's just my friend. That's my only reference as to who's done this paper. Okay, so what else do we go over? The calibration curve of glucose. I don't remember this one. Oh, okay, this is where it's the Benedict solution to identify the color. You compare it to a calibration curve of the absorbance using a colorimeter. That's like such a vague practical. I rarely remember doing that one. I think the general gist is the higher the concentration of glucose, obviously if a person is diabetic, it's gonna have obviously some level of glucose in. And you can talk about how if they have a high concentration of glucose, not all of that glucose can be reabsorbed in the nephron, and therefore it's in the urine. This is for AQA. I don't like Hardy-Weinberg. I'll be completely honest, I hate Hardy-Weinberg equations. Because I always end up overthinking how to do them. And I'll do some like wacky-ass maths to try and do it. Like, okay, I don't even remember the equation. This is bad. Hold on, it's it's p, okay, hold on. It's p squared plus, I think, 2pq plus q squared equals what? I think? Obviously, one of them is the dominant, one of them's the recessive. So that's going to be homozygous dominant, homozygous recessive. That's going to be heterozygous. I just kind of forget how to do it. Okay, that's it actually. I always forget that there's like that sub-equation of p plus q equals 1. That's why I always trip up, I think. Yeah, honestly, like, I know Hardy-Weinberg won't be worth, like, it won't be worth more than, like, 3 marks, I'm sure. Like, none of the math questions really ever are, but I'm hoping, at the very least, I can, like, squeeze a mark out of it, just for, improving. That's the thing, like, I want to ask, because me and Olivia were, like, really confused by this. I found paper 1 nice. I thought paper 1 was lovely. I don't know whether anyone else did, because Olivia apparently went online, and everyone was complaining about paper 1. I could not understand why. Probably because it was more knowledge-based than, like, application, but... Yeah. Honestly, I find paper 2 to be the hardest. I know, like, whoa, the grass is green kind of statement, but, like, yeah, I definitely find paper 2 to be the worst one. Okay, that, actually, yeah, like, it was odd that it was missing kind of nice things. Oh, you're doing A-level biology. Okay, man. buckle yourself in. It's not that bad. Like, it depends what topics you like. Cause, like, you know, if you're someone who loves the ecology stuff, you know, that'll go swimmingly. I hate ecology. I hate ecology so much. It's why, like, I suck with stuff like Hardy-Weinberg and things like that. What should you do to prepare yourself? Man, honestly, like, I think, year 1 of it is, paper 1 content is lovely. Like, biological molecules is, like, a breeze. It's a lovely, the year 1 content is lovely, the year 2 content not so much. It gets very yappy, but... I've never liked ecology. It's why I'm praying that generic questions don't come up. Like, I can explain primary succession, but I wouldn't be able to tell you what a population is, or, like, what an ecosystem is. Like, generic words like that, I just go through my head. Yeah, don't leave things to last minute. I mean, I've been very consistent with revision. Like, obviously, I know none of you guys, like, have seen this channel before, but the way I like to do revision is just by yapping about it and explaining it to other people. That's how I've always liked doing revision. So find a way that works for you. If you kind of start, like, you know, doing flashcards, but they don't help, focus on something else. Oh, natural selection would be lovely. And mutation, that would be a lovely one. Natural selection would be lovely, because that's literally just the idea of, you know, random mutations leading to advantageous alleles. Over time, the allele frequency increases, because they increase the chance of the organism surviving, reproducing. I want to check whether I've missed anything there, actually. I don't know whether they'd ask you specifically about the different types of selection. Like, obviously you have, I don't know why I drew it that big. You have, I think, 3 types from what I remember. You have directional selection, where one extreme allele is favoured. You have stabilizing, where the, like, I don't know how you would even describe it, like, the least extreme allele is favoured in a population. You have, I think it's called disruptive, where it's like the 2 peaks, and that's usually kind of what leads to speciation. Because obviously, if you have something like geographic isolation, then there's going to be different selection pressures in one environment compared to another. I would love a question on speciation, because it's so nice to explain. Like, talking about different selection pressures, over time there's no gene flow, and obviously you get 1 mark by default just for saying that they can't reproduce to have fertile offspring. Okay, what else? What else do we go over? Mutation, I mean, it depends, like, what they ask. Some shit on, like, tumor suppressor... My hair is such a mess, I'm sweating. Tumor suppressor genes would be, like, decent, I would say. It's just the idea of, like, methylation, which leads to the gene being silenced, or that it can't be transcribed anymore. Stabilizing is average. Okay, it's average. Directional, 1 extreme, disruptive, perfect. Okay, yeah. That's cool, that's cool. That should be good, I hope. Okay, you know what? Just because I'm gonna, like, kill myself if it comes up tomorrow. Photoreceptors. You have rods, cones. Cones, I always remember as just being the colour one, and that's it. But it's this idea of retinal convergence. The idea that with rods, you have multiple rod cells connected to the same bipolar neuron. This ensures that every, I don't know how you would even, like, describe it. It basically ensures that the threshold potential is always met. Whereas with cones, it's this idea that each cone is connected to its own bipolar neuron. Therefore, you have a higher visual acuity in cones. In rods, you have retinal convergence. So it's this idea of organisms that want to see in the dark would have more rods. Organisms that want to see hyper-detailed shit would literally have more cones. And that's literally it. Rods can also be called spatial summation. There's a lot of terms that, like, you can easily get mixed up on, like visual acuity, retinal convergence, summation, visual sensitivity. I don't say bio, you just came up. Oh, okay, I hope it went well. One of my friends did that, and he said it was okay. Supposedly, half of it was, like, a bit all over the place. This is for AQA. Yes, estrogen may come up. I don't... Thing is, I kind of... A lot of people in my class don't seem to like that one, but as far as I know, it's literally just the idea that, I mean, whatever. It's this idea that it can diffuse through the membrane because it's lipid soluble, it's a hormone. It's not a transcription factor, which is what a lot of people get wrong. It's this idea that it binds to the transcription factor, causing it to change shape so that it is complementary to the DNA. That's what I think. Oh, they did? Oh, man. Oh, thank you, thank you. Honestly, I did not expect this many people. Like, for context, with literally every other stream I've ever done, ever, I usually get a boatload of views after the stream itself. Which is kind of awkward, because, like, you know, I'm not a teacher. Like, it is a tad awkward, but it's chill. Like, I like doing these streams. I like yapping about stuff that interests me, and it just so happens that, you know, memorizing mark schemes and revision content is what interests me. Oh, that would be a lovely question. We've literally just done this today. Ow. That would be a lovely question, because I'm sure for one mark you could talk about the idea that Malignant are able to... I don't know how you pronounce... Is it metastasize? They're able to, like, break off and spread, whereas Benign remain localized. What are the other differences? Like, I know it's obvious, like, Benign, like, remains obviously localized. It's limited in terms of, like, growth. Yeah, okay. Slower growth, faster, okay. That would be a nice one. Like, the comparison questions are usually lovely, because even if you know one, like, you can kind of contrast it. Like, same with the rods and cons, or fast twitch and slow twitch. Like, you're able to compare both really well. Yeah, that would be a nice one. I like how we all say this, but, like, you know fine well they're gonna ask, like, some abysmal things tomorrow. You know they're gonna ask some abysmal things tomorrow. Like, the question on pairs for paper 1, like, that was just explaining co-transport, but with, like, 90 additional steps. I realised, actually, I made such a tragic mistake on paper 1. At least I think I did. That question where it was asking, about sucrose being a polymer, I put that it was, and I'm 90% sure it's not, because it's a disaccharide. Oh, that is cool. What a potential... Yes, that's what I put. Because, yeah, it's like the pH value, like, I think it's the decrease in the decrease in hydrogen ions is what raises the pH, I think. I don't know. I think the water potential thing was because, what was it? When sucrose diffuses in, it lowers the water potential, so then water would move in. It's just a weird one, though, because, like, you wouldn't... I thought we would get a co-transport one for, like, oh, explain it in the ileum, but no, apparently they hate us. I wonder whether the grey boundaries will be lower this year, because a lot of people found that, like, the paper one difficult. Again, not to be a number one glazer, but I didn't find it too difficult. The worst question, in my opinion, was talking about the adaptation of a leaf. Oh, the mycells question was lovely. Like, I don't know how many of you do this, where you look, when, I don't know how, like, your guys' schools work or whatever, but, like, they obviously make us sit in the exam hall for, like, 5 to 10 minutes before it starts, with the paper in front of us. I was just, like, staring through the paper to try and read what it was, and I saw my cells, and I got happy. Oh yeah, like it got worse. Oh, I realised there's the adaptations in... Wait, no. Is that paper 1 or 2? Oh, ghost crabs are cool though. I googled what they were after the exam and they're kind of fire. The adaptations of plants meant to conserve water. Is that paper 1 or 2? That's paper 1, right? Because transpiration? Oh god, hold on. Which one is it? Because I low-key just... I'm panicking now. Okay. That's fine. Because I was going to say, that's the one thing that I always forget. But even that's not that bad. It's just like dimples in the stomata, reduced surface hair. Wait, that's another one. Hold on. I'm pulling up this spec. I want to see whether I've missed anything. Okay. Photosynthesis, respiration, energy and ecosystems, nutrient cycles. Yes, I was so disappointed, but according to my teacher, that means that it's probably going to show up in paper 3 in some capacity. Also, it could appear in paper 2 as like, you know, a question about the gene technologies, like, you know, we did get a question on bacteria and the plasmids. I guess you could argue that was somewhat immunology, but not really. Nutrient cycles. Phosphorus cycle would be an interesting one. I would argue if phosphorus or nitrogen cycle came up, it would be at the back, right? Like, you know, as that comprehension question. Which would be annoying, because I hate that question. Oh, I'm praying that water comes up as the essay question in paper 3. I would please. That would be so nice. Because I know exactly what I would talk about. Like, you could talk about transpiration, translocation, and the mass flow hypothesis. You could ramble about just the properties of water. You could ramble about osmoregulation. That would be kind of what I would go for. Cycles on movement. Cycles would be like an odd one. I guess mitosis, nitrogen cycle, phosphorus, meiosis maybe. I'm trying to think now. What else? Oh. Yeah, I haven't actually. Oh, obvious. Oh my, why did I? Come on. You know what it is? It's that essay question. I forget everything. I forget everything for the essay question. Like, it could ask me, explain the importance of ATP in biology, and I will forget everything. Like, it took me maybe 10 minutes to come up with, 4 topics. Which is so, I gotta get better. Calvin cycle, though, would be lovely. Talking about Krebs cycle and link it to oxidative phosphorylation. Yeah, that would be lovely. I hope we just get some generic questions on things like glycolysis. Because, again, glycolysis could come up for 4 marks, and all you need to say is phosphorylation of glucose, reduced NAD, leading to 2-pyruvate, via oxidation of triose phosphate, net gain of ATP. For 4 marks, it's lovely. Oxidative phosphorylation, I feel like I know that well, but I probably wouldn't get 6 marks for it. Yeah, nitrogen, the role of nitrogen. Oh yeah, we need knowledge questions. Realistically, that's what we're all good at. There is no need for us to get it. But again, I mentioned this at the very start of the stream when, like, no one was watching, but I'll quickly go over it again, because I don't know how many people in my class, like, pay attention to it and stuff like that. But it's something, like, I noticed in terms of the mark schemes. For the evaluation questions, the ones where they give you, like, some wacky-ass data, and they ask you to evaluate a conclusion, you can, like, basically use the same buzzwords every time to get marks. Like, one thing is the overlapping of standard deviations and the error bars. If that appears, you can always refer to that. If they overlap, obviously that means that the data is not statistically significant. If they don't overlap, it's going to be, you know, significantly different. In terms of evaluation questions, look for things like how many species. If they've only done the experiment on one species, but they're making a general statement, talk about that. I saw one question where they did an experiment on mice, and then they asked you, oh, this treatment works for humans. Like, no, like, that's not how it works. So if they've only tested it on one species, talk about that as being a limitation. The easiest mark you can ever get, is talking about statistical tests, right? Literally, statistical tests, 9 times out of 10, they won't have done one. So mention that. Say that they haven't done a statistical test to see if it's significantly, you know, important. Trends, yes, that is good actually. Misleading, sample size, yes. Also, like, location, like, if the experiment's been done in, like, one forest, they can't make a general claim about it. Like, and that was, that's what they did in the crab question, I believe. They only tested one beach. They didn't use, like, a good amount of crabs. Like, they did, like 6 or something for one, and then, like, 2 for another. Like, yes, this is AQA. Bro, that is the thing. Like, they're gonna give us some weird-ass questions. Especially for topic 8. Yes, that is a good one, actually. The idea of, like, if they're doing it with participants, just giving general, like, context like that. Obviously, the positives, you could say, if they've been randomly sampled. Like, if it says, oh, there's 100 participants, they were randomly put in groups A, B, and C, that would be a big positive. You could get a mark just for talking about random sampling. I'm trying to think what else. Obviously, just like the misleading data, which they did ask in the 2024 question, the final question right at the back. They had a graph where it showed, like, a very weak correlation, and then they zoomed it in, and it was completely different. Obviously talk about the scaling and how it presents false data. That's another one. Correlation does not imply causation. That's just a lovely mark point. Like, if you're being asked to explain data, that one can be a little bit annoying, but if it's a very obvious, like, scatter graph, talk about a correlation. Like, 9 times out of 10, you'll get a mark just for saying that. Grade boundary, man, honestly, grade boundary inflation is like... Last year, I have a spreadsheet that I made to look at the grade boundaries. They are lethal. Last year, they were at an all-time high. I am begging. Oh, is it? Honestly, it's because I've seen that in mark schemes. That's the reason I put it. But if I'll take that. Don't worry. Honestly, I think that's more of a last resort thing anyways. Like, I wouldn't put that unless I literally was like, I have no idea what else to put. I would definitely focus more on statistical test, the species, things like that. That's more of like a literal last resort, if I have no idea what to say. Oh, it is? Oh man, I must have, Huh. Like here, this is gonna be like 2006 level budget here. I apologize. Like, that's the spreadsheet that I made. I know it's flipped, but literally, you can see 2024 had the highest boundaries, which is like kind of shit. Like, for context, an A star, in 2018 was 166 total marks. Last year, if you got 166, you would have just scraped an A. Which is like, that's lethal. You would have gotten a C in 2024 if you got an A in 2021. Like, that, I think that just shows, like, at the end of the day, even if on results day you get, like, you know, you don't get an A or something, it's a grey boundaries. Like, it literally is. The grey boundaries fuck everything. You could be a handful of marks off, like, an A star, but they're just pricks about it. Okay, well, it's almost 8. So I'm probably gonna stream for, like, 20 more minutes, and then I'm gonna go chill. That is literally it, because as time goes on, more revision material becomes available. The mark schemes begin to get, like, understood more. Oh, no. Oh, man. Okay, that is good, though. If it goes bad, honestly, an A is doable. Like, the way I have it on my spreadsheet is, on average, I think it's 70% for an A star, 60% for an A, and 51% for a B. And while that might seem a little, like, mad, I don't think it is. I really don't think it is. I think for a 60%, if you think about it this way, you're always going to get, like, you know, those easy mark questions where, like, you're having to explain a nice process. They don't really... Yeah, exactly. Like, it's annoying. They sometimes will reuse questions, but in, like, slightly different veins, but I definitely see what you mean. Like, I don't know whether you guys saw, but there was a question last year on explaining what, I think it was a population, or it might have been a species, explaining what a population is. And usually that's a 1-mark question for saying, you know, it's a group of the same species living in the same, like, area. But you also then would have had to add the whole idea of reproducing to have fertile offspring, which was never a mark point before. So it's this idea that they're literally upping pre-existing questions with filler just to get more marks out of it, because they know what people are going to put. Which is, like, it's so deceiving. Oh, okay. You need ABB. Man. Oh, physics. Honestly, our school doesn't even do physics at A level because nobody picks it. I'm convinced there's one person that does it, but it's only because they came from another school that already did it. So, man, I have not heard about A-level physics, but judging from my experience with GCSE physics, I'm assuming that it was horrible. And you have my deepest condolences. Because, yeah, you would not catch me dead doing physics. Oh, further maths. I'm trying to think how many people in my school... I think maybe 4 in my school do that. Supposedly the papers went okay, though, for AQA. I think it's AQA anyways, it does further maths. Oh, man. That is rough. Okay. I want to just quickly, like, wrap my head around stuff. Okay. So, the energy transfers topic. That's everything from photosynthesis, respiration, the nutrient cycles, things like that. I think that's, like, the safest topic. The practicals you can get asked for that are probably going to come up. The respiration practical is probably going to come up. Actually, no. Because I realised they asked that last year, so maybe not. Photosynthesis, most likely going to come up as a section. I would love a question on the chromatography practical, because it's so, like, basic. The DCPIP practical isn't even that bad, though. You know what would be a bad one? Is a question on IAA and plant growing factors. That would be a bad one, because they usually ask that in really weird context. You know, like, one year where it was, like, using bioassays. Yeah. I think that's a thing, though, is, like, both of them probably won't come up, purely just because they're very, like, similar. But there are nice questions you could get asked if they do come up. Like, oh, how would you modify it so that it's testing for CO2 output instead of, you know, oxygen uptake or something like that. In terms of practicals for the organism's response, I would cry a river of happiness if we got a question on simple organisms responding. That would be lovely. You know, taxis versus kinesis. You could really ramble. You could ramble so much about that one. The other practical for that one is the glucose one, I believe. And honestly, that one isn't too bad. It's this idea that if a person is diabetic, then they are going to have... The Benedict's test will show that they have some, like, glucose in their urine. Then you can obviously link that to the idea that because of the impact of diabetes, you can waffle about that, it means that not all glucose can be reabsorbed into the bloodstream during selective reabsorption. Then we have genetics, populations, and ecosystems. A quadrat practical would be lovely, but I think they asked that last year, if I'm not mistaken. I would love one, but a question on succession would be lovely, talking about pioneer species, changing the abiotic factors, environment becomes more suitable, that would be lovely. Speciation would be lovely too, because it's, even if you get asked about geographic, you can really just link it to the other one, like, really nice. Different types of competition. I see they've asked that one, like, somewhat frequently. Intra-specific and inter-specific. Inter is between different species, intra is between the same. Yeah, I would love... I would love a quadrat question. Then I do believe there are no questions for, or practicals for gene expression, because that whole topic is literally just mini practicals at the end of the day. It's like restriction endonuclease, in vivo cloning, which is the one where
you use restriction and nuclears, you add a load of promoter regions, you cut up a plasmid. Honest, I would like just hang myself if I got a question on that. Like, yeah, no, I just count me a fail. Yes, that would be lovely. Mark release recapture would be so good. That one is really. That one is. I think the mark scheme for that one is really lovely. As far as I remember, it's literally just capture mark release, leave time for the species to disperse before the second sampling, and then it's just basic math to calculate the estimated amounts. Then obviously you could get asked questions like, oh, why would you wait until the population's dispersed? What would you do to make sure that they're unharmed when you're tagging them? That would be, make sure that whatever you're tagging them with doesn't increase the chances of predation or death. In terms of everything else, chi-square testing, I have this one written down purely because I hate it, but that's literally just observed over expected, squared, divided by expected, and then you add it all up. That shouldn't be bad. Hardy-Weinberg principle is where there's no mutation, no migration, no random mating, and it's that genetic variation will remain constant. It's in equilibrium. Genetic drift, I've just done this today. It's an annoying one to explain. The way I remember what scenarios to use... Oh, honestly, yes, I don't know a thing about that. Chi-squared... Okay, hold up. Chi-squared, I believe... I mean, it's kind of like in the equation, observed over expected. It's whether the thing that you're seeing is actually what you're meant to be seeing. Yeah, Spearman's rank is correlation. I would love Spearman's rank, but that came up in paper 1, so... Yeah. The student t-test, I don't remember that one in the slightest. I've rarely seen questions on that as of recent, unless I'm mistaken. I don't even remember how you do it, which might be a problem. I'm gonna look at that quickly. I want to see if there's any notes on that, but I've done. I love when the Wi-Fi just stops working. I think literally you could just say to determine statistical significance. I think that's really what you could say for a mark. Like, obviously, the chi-squared is going to be for whether the results you're seeing are what's expected, which, for example, for things like dihybrid crossing, whether what you're seeing in terms of the offspring is actually what you're meant to be figuring out. I just graduated next year. Buddy. Oh yes, my friend has sociology paper 3 tomorrow. Man, that's the afternoon though. That is nasty, to be honest. You have my condolences. Oh man. But this is a thing I hate with exam season, is like how many things overlap. Like I've had history paper 3 today. We did ours on the witch craze, and oh my god, like I'm fucked with history, but yes, degrees of freedom. That is, as far as I remember, degree of freedom, I'm pretty sure... No, there's another thing. No. Isn't degree of freedom where it's literally just like the number -1? And that's how many degrees? I remember the p-value. If it's greater than 0.05, I don't recall what that means. I don't even remember. I get it mixed up. One of them means the data is statistically significant, one of them doesn't. And I don't remember which is which. Yeah, that's a thing as well. Because for chi-squared, if that value is greater... Oh, what the hell? Right, hold on. I have it written down here. If the chi-squared... If the critical value is greater than the chi-squared test, then you accept the null. Do without what you will, I have no idea. If it's less than 5%, okay. I think that's good then. You can always get a mark literally just for saying accept null, reject null. Like, that's always a mark point. If p-value is greater than 5, it's likely it was obtained. Okay, thank you. So if it's greater than that, no. Okay. Oh yeah, we are all cooked. Like, let's be real, guys. We're going in this thinking like, oh, we're gonna get a lovely question on photosynthesis and shit like that. Like, what just get the most abysmal questions? Thing is, I thought paper 1 was lovely. Everyone else hated it. Maybe that means this paper everyone will like? I don't know. Very quickly, just because I know I'm going to forget it, the light-dependent reaction is in the thylakoid membrane. Calvin cycle is in the stroma. Which I forgot the name of. I don't know whether you guys... I don't know whether it was the right answer. The question where you had to label the parts of a chloroplast, I forgot what the name of the stroma was, and I sat there for 10 minutes when I finished everything trying to remember, and I couldn't. And it actually killed me. Yeah, it was, to be fair. Like, it could have been way better. Oh, really? I have it written the other way around. Oh, shit. Because I have it that light-dependent is the thylakoid membrane. Because obviously it's basically like oxidative phosphorylation. I have Calvin cycle in the stroma. Could be wrong there, could be wrong. Then I have glycolysis cytoplasm, which is true, because... Oh! That just reminded me. Respiratory substrates. Oh, okay. My bad. I was confused for a sec. Respiratory substrates. I don't remember this. I think it's lipids, like triglycerides, get broken down into glycerol, and I think that goes into the glycolysis, as far as I remember. Oh, okay, no, I need to go over this. What the hell? Oh, did I? Oh, I meant to put LIDR, like independent. I'm so... Hold up. That's not coming up on my stream chat. What the hell? Fatty acids from hydrolysis of lipids converted to acetyl acetyl coenzyme for Krebs, I think, if I'm not mistaken. Amino acids is Krebs. Okay. praying there's no, like, free marker on that then, because I'm fucked. Yeah. Let's hope. Oh, man. Honestly, like... I don't know, guys. Like, okay, the thing that, like, slightly uplifts me about all of this is... I don't know, actually. I was gonna try and give, like, some motivational ass, oh, like, it's gonna be... Yeah, I have no idea. I have no idea. Okay. What I'm probably gonna do is wrap things up here. Honestly, thank you guys so much. Oh, the only thing I remember is, I thought glycerol, was used in glycolysis somehow. I don't remember. I'm gonna revise that. But that's all I'm gonna wrap up for. That's all I'm gonna do. I realize I'm not ignoring people. I am very sorry. On my laptop, it's not coming up with half of the chat messages. And that's really weird. Okay, there, I fixed it. My bad. When there's no glucose... okay, yes. That's what I thought. I think? Yeah, that's what I assumed in anaerobic. Honestly, at this point, it is what it is. But look... thank you, like, seriously, guys, thank you so much for watching. I literally, when I started this at, like, 6:30 or whenever, I was like, yeah, Luke Kelly, who's the mod with the pinned comment, Luke Kelly will probably come on, say one thing, and then that'll be it, and I'll probably just be yapping to myself, which is what I do for all of these streams. I like learning things just by yapping about it. But I never expected to get over 350 people watching. That is amazing. Thank you guys so much for watching. Seriously, good luck with the paper tomorrow. I know I gave that whole thing about being cooked, but I think the thing is, the fact that you guys are, like, literally revising now and stuff shows that, like, you're more committed than most. We should be okay. We should be. I don't know. We should be. But yeah, honestly, good luck everyone. I will be continuing to stream over the next few days for paper 3, which it'll be more detailed. I'll be going over past papers, stuff like that. Please don't feel pressured to, like, subscribe, like the stream, anything like that, because once A-levels are done, I'm just going to be uploading random videos and stuff. But if you guys do want to come back, please feel free to. But yeah, good luck everyone. Take care. And yeah, honestly, I don't know. We'll see how it goes. Best of luck everyone, and take care. Thank you.